We will talk about drug-induced single-cell transcriptomic landscape is revealed by pathway trajectory analysis with tensor imputation. Understanding the complex responses of human cell systems to drugs is vital in medical and pharmaceutical research. Genome-wide identification of the transcriptomic responses of human cell lines to drug treatments is a promising approach for this purpose. Drug-induced gene expression data have been utilized in a variety of analyses. Recently, single-cell gene expression profiling becomes a powerful tool for understanding cellular systems at the single-cell level. However, it is very difficult and expensive to experimentally observe drug-induced gene expression profiles for all cells. Drug-induced single-cell gene expression data can be represented by a tensor structure. For example, a drug-induced single-cell transcriptome dataset comprising drugs, genes, and human cells can be represented by a third-ordered tensor. However, most parts of the tensor are missing or unobserved. We have two objectives in this study. We propose a novel computational method, tensor-based imputation of gene expression data at the single-cell level, tigers, to predict unknown parts of drug-induced single-cell gene expression data. To evaluate the usefulness of the proposed method, we identify the transient of drug activity at the pathway level. We analyzed drug-induced single-cell gene expression data reported in previous studies. For example, a pancreatic islet dataset can be represented in this way. This dataset has a large number of missing or unobserved values. We evaluated the performance of data imputation using RSE values. The lower the RSE value, the better the performance. This result suggests that tigers show the best performance. Next, we evaluated the performance of data completion in terms of cell type classification. These figures show the scatter plots of cells obtained after applying UMAP to gene expression data with and without imputation. In the top panel, each gene expression profile is colored according to the cell type, and in the bottom panel, each profile is colored according to the drug. As shown in the observed data, almost all cells from the same cell type tended to cluster together. This result suggests that tigers can recover cell type-specific gene expression patterns. To evaluate the biological validity of the imputed values, we checked gene expression values for pancreatic marker genes. In pancreatic islets, insulin is known as a marker for beta cells, and glucagon is a marker of alpha cells. Each density plot is colored according to the cell type. For example, insulin and glucagon were highly expressed in beta and alpha cells respectively as expected. For cells imputed for all drugs, tigers with TT decomposition could predict cell type specific gene expression. Therefore, these results suggest that tigers with TT decomposition can predict potential gene expression for all drugs even if a cell is not treated with the other drugs. These heat maps show the results of pathway enrichment analysis for drug-induced single-cell gene expression signature of an antimalarial drug, Artemether. The top panels show the activated pathways, and the bottom panels show the inactivated pathways by Artemether. The left panels show the result based on observed data, and the right panels show the result based on imputed data. These results suggest that the detected regulation of pathways is different before and after imputation. For example, the significant inactivation of cell cycle pathways in alpha cells and that of the interleukin-17 signaling pathways in beta and ductal cells were detected after tensor imputation only. It has been reported that the IL-17 family may be involved in cancer development by promoting chronic tissue inflammation. Thus, the inhibitory effect on the signaling pathway might be biologically relevant. This result suggests that the proposed tensor imputation method enables the discovery of a different set of biological pathways. We proposed to analyze the regulation of biological pathways along the inferred cell trajectory. We identified the impact on pathway regulation for each vertex on the inferred trajectory. As a result, we can explain an inferred trajectory from a branch node to a leaf node in terms of the impact of a drug on biological pathways. Therefore, the pathway trajectory analysis provides information on the transient effects of a drug along the inferred cell trajectory. The left figure shows the activated pathways for Ultimeter, and the right figure shows the inactivated pathways. Each row indicates a point on the inferred cell trajectory. Black elements in the matrix on the right side of each heat map indicate the correspondence between cluster numbers and the nodes on the trajectory. For example, leaf node 8 and 9 are on the cluster 1 in the UMAP. Nodes in the same cluster tended to have similar pathway regulation. However, there were some exceptions. Therefore, the pathway trajectory analysis makes it possible to identify drug activity along the inferred cell trajectory and to discover potential relationships among different clusters. We summarize this talk here. Thank you for your attention.